So in my last video, I talked about how protein structure determines protein function. And in subsequent videos, I'm going to ask the question, where does protein structure come from? In this video, I want to just focus on the question, what is protein structure? And so what we're actually going to think about it is as a tour of protein structure. So proteins are built out of amino acids, which are molecules that are characterized by having an amine and a carboxylate joined by a C alpha carbon in the middle. At physiological pH, the amine is protonated and positively charged, and the carboxylate is deprotonated and negatively charged. Every amino acid has these characteristics, and then they differ from one another in their R group, which is a variable group that determines the identity of the amino acid. Now, another way that this is often referred to is in sort of collective blocks. So we would say this region of the amino acid is the backbone, whereas this region of the amino acid is the side chain. So the backbone is the same between every amino acid. The side chain is what's different. If we look at two amino acids, we can see this operating in play. So if you've never seen a molecule before, you'd still look at these and say, wow, they look kind of different, right? The one on the left has a bunch of stuff hanging off of it, the one on the right doesn't. But if you look more closely, you say, yeah, but you can see they have their commonality, right? They both have the amine. They both have the carboxy, right? So they have the same backbone, different side chains. Now I'm going to do an entire video on the amino acids. So for the moment, I'm just going to say that proteins, naturally occurring proteins, have 20 different amino acids. These differ in their side chains in interesting ways. So we've got an alanine here, which just has a methyl group. We've got that tryptophan, which is the other amino acid I contrasted it with, which is really big. There are other ones that have like a negative charge on the glutamate or a positive charge on the lysine. But overall, if we look across amino acids, we've got this sort of variety of different side chains attached to an identical backbone. Now, amino acids can be joined end-to-end via peptide bonds. And what a peptide bond involves is taking the carboxy end of one amino acid and the amino end of another and removing a water, dehydrating to create the new bond. And we can tell it's a dehydration reaction just by accounting. I look at that peptide bond and I say, well, what am I missing in that peptide bond from before? Well, I'm missing an oxygen from that carboxylate. I'm missing 2 protons. That means I had to lose a water somewhere. A protein, also known as a polypeptide, is simply a chain of amino acids that's joined by peptide bonds. So here I'm showing 4 alanines that are just being joined end-to-end by peptide bonds. The only reason I stopped is because I got bored with continuing to add more because this is this open-ended chain, right? I could just keep sticking amino acids on end-to-end. Now there are really 4 important features of this chain I want to highlight. The first is that this chain is linear, not branched, right? It's just a single linear chain. There's no branching happening like you might see in, say, a carbohydrate. The chain is open-ended. It can go on forever, right? There's still a new place to add another amino acid after I add the next amino acid. If I add another amino acid, there'll be another place to add a new one. It's the amines and the carboxylates. It's the backbone, not the side chains that they're making these. And that basically means I can add any amino acid, right? There's no amino acid I'm going to add that's going to prevent the chain from growing further. And finally, the 2 ends of the chain are different, right? I've got a positively charged amino and a negatively charged carboxy terminus, and these are different. We can tell basically which side of the peptide we're looking at based on which chain is there, or which group is there. And I think a good physical analogy for this is actually brio trains. I don't know if any of you grew up with them, but I certainly did. And what brio trains are is they're wooden train cars that are joined by magnets. So there's a north end of a magnet, a south end there, a north end, a south end, etc. And this is a really nice analogy for proteins for a variety of reasons. First of all, there's directionality to it, right? I can put this train car in this way, but if I've tried to put it in the opposite direction, it would actually repel. It wouldn't stick, right? So I have to put it in a particular direction. So there's the directionality of the chain. There's the fact that the chain can go on forever, right? The only thing that limits how many train cars I can add is how many train cars I have. Other things that make it similar is it's all of the interactions are being driven by the backbone, which is identical with every train car, yet they can have different side chains, right? This one is a square, this one is a circle. So they have different side chains, even though they have identical backbones. So when you're trying to think about like, what is a polypeptide? What are its features? Just think about what are the features of a brio train and you'll be well on your way. Now, the first piece of structure we can get out of an amino acid is actually visible even here, or sort of out of the first structure we can get out of a protein is shown here, even with these train cars, and that is the primary structure. It's just what is the sequence of amino acids that we added end to end? What is the sequence of the train cars? We can see a real example of a protein's primary structure right here. This is the primary structure of a protein known as staphylococcal nuclease. It starts with a lysine, goes to a leucine, goes to a histidine, goes to a lysine. That is its actual amino acid sequence. We can take this sequence from letters and turn it into chemicals by actually drawing it this way. So I've now drawn the lysine with its amino terminus here. It's our carboxy side here. We're now going to add a leucine, a histidine, and a lysine. And hopefully what you appreciate is we're just adding this in a linear chain, right? We're just adding, there's the first peptide bond, there's the next peptide bond, there's the next peptide bond. But at the same time, while we're forming these peptide bonds, we have different side chains hanging off the side. So maybe compare this leucine here to lysine. Even though, you might not know the details of your amino acid yet, you can look at that and say, yes, that is different than this one. Now, to help with the visualization I'm about to do, I'm going to actually expand all of the spheres to their full size. So now I'm showing this short polypeptide as a set of van der Waals spheres. And now I'm going to keep adding on amino acids. So I'm going to add the glutamate, the glycine, the alanine, etc., all the way out to the 140 amino acids that make up this amino acid chain. And hopefully what you see is you're like, wow, that's a long thing, right? It's a really long chain. Now, this is not the structure that this protein takes on in a cell, right? Proteins don't tend to be these compact, unfolded structures most of the time. Rather, this folds down to a very specific compact structure. So what I'm showing here is it's sort of winding itself up to its final protein structure. Now, I have to put an aside on that, and this is a non-physical mechanism of protein folding. The protein doesn't sort of wind up like a ball on a string. Rather, the whole sort of chain collapses all at once onto itself. But still, I like that animation because it shows you, yeah, this really is the chain wrapping up on itself. So take away that from that when a protein folds, it's the chain wrapping up on itself into a very specific structure. So here we're seeing the final structure of staphylococcal nuclease. It's this very specific compact structure like most proteins form. What I'd like to do now is start changing how we look at this protein to go from this, which is kind of the most physically motivated representation of this protein, to the most abstract version of how we draw this protein. So here we're seeing all these atoms that are packing together. It looks kind of like a blob. This is basically how a cell sees the protein. But I don't know about you, I look at that and I say, there's a big blob of protein. I don't know how to interpret that. So let's figure out how we can draw it in a way that's maybe more useful for our human brains. So the first move I'm going to make is actually to make it look worse. I'm going to take all those big van der Waals spheres and I'm going to shrink them. And when I do that, what I get is, I have no idea what I'm looking at anymore. But we're going to, trust me, we're on the way to something good here. Okay, the next move I'm going to make is I'm going to just erase all of the side chains. So they're still there in the structure, but I'm going to get rid of them so I don't have to look at them. And when I do that, now maybe we're starting to see something that looks a little bit more reasonable. I'm starting to see, there's a chain structure in there somewhere maybe. I'm going to highlight that chain a little bit better now by drawing a big gray line between each of the C alpha carbons. Remember, each amino acid has a C alpha, so it's going to go C alpha 1 to C alpha 2 to C alpha 3. When I do that, now we're starting to get somewhere, right? Now I can start to see, oh, look, there is a chain that actually I can trace all the way through the structure. And the other thing I start to observe here is there's a lot of carbonyls and amide protons hanging off of the side of this thing, right? So I've got all these carbonyls, oxygens, hydrogen bond acceptors, and all these protons, hydrogen bond donors, hanging off the side of the structure. That suggests hydrogen bonds. So if I turn on hydrogen bonds, I suddenly can see some really interesting features, right? So here on the bottom, I've got this stair-steppy looking thing here. I've got another stair-steppy looking thing here. And then at the top, I'm like, oh, there's some kind of plate, right? I can see how the protein is folding up on itself and forming a bunch of hydrogen bonds. Now, this representation, I think, is a really nice way to look at proteins, but it's still kind of visually cluttered. So if you look at a textbook or you look in a paper, people will often go one step further and they'll draw it in an even more simple, abstract way. Here's what it is. This is the cartoon representation of the protein. And while maybe not totally realistic, it's quite pretty and it's quite easy to see how the chain moves. So here's that stair-steppy thing down here. I now represent it as this beautiful helix. I've got this stair-steppy over here. It's another beautiful helix. And that thing I call the plate in the middle, that's these arrows, is forming this nice beta sheet. And so this cartoon representation is a very common representation of proteins, very helpful, but also keep in mind that it's pretty abstract, right? What the protein actually looks like to the cell is a big globby thing. Okay, What the cartoon thing, though, does really nicely illustrate is that there are three types of structures that actually tend to occur in proteins that are defined by the types of backbone hydrogen bonds they make. And these are alpha helices, beta sheets, and loops and coils. Collectively, these are known as the protein secondary structure. So recall the primary structure was the amino acid sequence. The secondary structure are these elements that I called the stair-steppy thing in the plate. They're these helices and these sheets. So what we're going to do is just go and look at these in different protein examples and sort of understand their features a little bit better. So first, alpha helix. Alpha helix is a piece of secondary structure that's forming hydrogen bonds between amino acids nearby in sequence. In this representation, I'm showing every peptide bond with a sort of pink square. And then I'm showing the backbone peptide or the backbone hydrogen bonds as these yellow dotted bonds. And hopefully what you can see is it's sort of this nice array of hydrogen bonds. To illustrate how near this is in sequence, it might be helpful to actually watch this sort of collapse in on itself. So now I've stretched out an amino acid sequence or a polypeptide into just a chain. Now I'm going to let it wrap up on itself. And hopefully you can see, oh, it's just a stair-step climbing, right? That's what the helix is. It's this stair-step formation of bonds. And it turns out there's sort of a characteristic step to this. So what I'm going to do is we're going to look at this video. I'm actually going to zoom up on it. We're going to look at this video at the bottom. And what we're going to ask is, this amino acid here, we're going to call this amino acid 1. What is the amino acid to which is, actually let's go down here, this will be easier to see. This is amino acid 2. Which amino acid is it bound to here? So we're going to count as it goes by. So ready, oh, apparently I can't do it from there. All right, so there it goes. It just went by. 1, 2, 3, 4. That's where it goes to. I don't know if that was totally clear, but basically, it's an I+4 rule. If you ask where do the hydrogen bonds form, it's every fourth amino acid backbone is forming these side chains. It is a characteristic feature of alpha helices. All alpha helices follow this hydrogen bonding rule. Because of this, alpha helices are actually very rigid structural elements. And imagine for a second we're trying to bend, going backwards, we're trying to bend this protein, or bend this polypeptide this way. If we do that, what we're going to be doing is we're going to be stretching all the hydrogen bonds on this side, and we're going to be compressing all the hydrogen bonds on this side. Polypeptide or alpha helices resist that, so they end up being these sort of rigid rods from which you can build protein structures. And in fact, there are proteins that are basically all alpha helices. So this is actually a steroid receptor, and what you'll notice is it's just got these giant alpha helices that are all packing against each other, forming this nice, rigid, functional molecular machine. Okay, so that was alpha helix. What about beta sheet? So beta sheets are a little bit more loosey-goosey in their rules. What they are is they're formations of hydrogen bonds between the backbone, yes, but there's no simple rule of like, oh, it's every fourth amino acid. They can be from things distant in sequence. In this protein, for example, you can see that there's this sort of long array of solenoid of things that's forming these hydrogen bonds. Excuse me. These beta sheets come in two flavors, parallel and anti-parallel. So this beta sheet here is anti-parallel because the two sheets that are coming together have different directions, right? So this is the N terminus over here going up to the C terminus or the amino terminus to the carboxy, and then it's interacting across the way with a carboxy going to amino, amino going to carboxy. You know what I mean? They're going in opposite directions. You can contrast that with this side where the two chains are parallel to each other. So a beta sheet can be either parallel, anti-parallel, or parallel, and you can notice this whole sheet is actually a mix. It starts out anti-parallel and then it becomes parallel. The superpower for beta sheets is forming large interaction surfaces. So this is actually three proteins that are interacting with each other, which I've colored pink, green, and blue. What I'm going to do is go through a set of moves where we're going to take the blob, again, this is what the protein looks like to the cell, and we're going to put the cartoon in so we can see how the components interact. So first I erase the green and blue, and we just go to the pink. We rotate, so we're looking at the interaction surface. We turn it into this cartoon representation, so now we can see this as, a cartoon, and then we flip back and look at it from the top. And hopefully what you appreciate from that set of moves is that beta sheets are forming these big interaction surfaces where this thing can pack together. So it's one of the big superpowers of a beta sheet is to form interaction surfaces. Now those interactions actually don't have to be just between proteins. I thought this was a fun enough example, I just had to add it. This is a protein that is produced by the spruce budworm, which is a pest that's causing all kinds of problems in Canadian forests. And since it lives in Canada, one of the big problems that this bug faces is it freezes, right? There's a lot of cold that it has to deal with. So it makes this protein here, which binds to ice. So you'll notice there's this array of hydroxyls on it. Those are the amino acid threonine. Turns out those are spaced exactly right to interact with the lattice of frozen water molecules, which means when an ice crystal starts to form inside of the spruce budworm, these proteins can actually bind to it and prevent it from growing further. So it actually literally prevents ice growth, thereby protecting the spruce budworm and being bad for the Canadian spruce. Okay, so there's these three structures. We've talked about alpha helices, these rigid rods, beta sheets, these flat interaction surfaces. The last one I want to mention are loop coils. Loop coils are, we've actually already seen them. They're basically the bits that connect the other pieces of secondary structure. So everything in orange here is a loop or coil. You might think, oh, it's kind of boring, just the connector bits, but it turns out it's really important. So here I've taken a protein that's the same length as an amino acid sequence, the same length as a real protein, and just made a giant alpha helix out of it. And what you'll notice is it's just a giant line, right? It's not really doing anything. It's just like a giant helix. If we want to turn this into a real compact structure, we need loops, right? So this is what all these loops do. So this amino acid sequence here is the same length as this other one, actually. But it now can be much more compact. We can see it much better as a sort of single object because the loops broke up that helix, the helix, to make it into a single object. Additionally, loops tend to be flexible regions of proteins. So this is the protein I showed earlier, staphylococcal nuclease, where now I'm showing the helices, the sheets, and the loops colored by pink, blue, and orange. And what I did is I ran what's called a molecular dynamics simulation. So I did a physics-based simulation of the protein structure and then took snapshots out of that and overlaid it onto each other. And hopefully what you can appreciate there is things like the helices are, I'm going to actually pause it so we can look at, oh my goodness, stop. Hopefully what you can appreciate is like the helices and, come on, there we go. You can appreciate is that the helix is pretty, it's not fluctuating very much in contrast with this loop, which is flopping all over the place. And it turns out in this end, this protein that's important for function, it kind of acts like Pac-Man when it breaks up, it cuts nucleic acids. That floppy loop is really important for the function. Okay, so what have we said thus far? Proteins have an amino acid sequence, a primary structure. Then they have a mix of secondary structure elements, which are alpha helices, beta sheets, and these loop coils. These secondary structure elements then assemble up into the next order of structure, which is called tertiary structure. So we've already seen some of these, but I'm going to highlight them here. So there's our antifreeze protein from a spruce budworm. It's a collection of beta sheets and loops. We've seen this protein here, which is an entirely alpha helical protein. This is used for transporting things across bilayers. We have staphylococcal nuclease, an enzyme, which is a mix of alpha helices, beta sheets, and loops. And then you can think of any other arrangement of structures here. But this whole idea of arranging the secondary structure elements into some three-dimensional structure is known as the tertiary structure. So primary is sequence, secondary structure is alpha helix, beta sheet, coil. Tertiary structure is how you pack those secondary structure elements together. Okay, we are almost done, but we have one more level of structure we need to talk about, and that is the level of quaternary structure. So I already showed this where we had three proteins that were sticking together in a defined way. When you have more than one tertiary structure sticking together, you end up with a quaternary structure. So unlike many things in biochemistry, at least this is sane and logical, right? We go primary, secondary, tertiary, quaternary. Quaternary is the assembly of multiple tertiary structures. This can be really important for protein function. I actually showed this in the last video. Remember we talked about actin filaments. So these are actins that stack on top of each other and then they can branch using this other molecule ARP23. That stacking branching ability to assemble into a giant quaternary structure is really important for allowing cells to say move with their actin network. So quaternary structure, let's just we're making a big protein, what do we do with it? It can actually have some really important functional roles. Okay, so we're just about done. I just want to summarize really quickly. Proteins are made out of long chains of amino acids joined by peptide bonds. This is the primary structure, whatever the sequence is. These chains form alpha helices, beta sheets, and loops. This is the protein secondary structure. Secondary structure packs together to form tertiary structure, so some specific three-dimensional conformation of the protein. And finally, proteins tend to form or can form complexes with other proteins, and when this happens, it's known as the quaternary structure. Okay, so that's all I have on our tour of protein structure. In the next few videos, we're going to ask, well, where does this secondary, where do all the protein structures come from? So I hope you join me for that, and thanks for watching.